My name is Nick. I'm a bioinformatics PhD candidate in the Boutros Lab, and today I'll be talking about my work in prostate cancer structural variant GWAS. So to start with some motivation on why should we study prostate cancer in the first place? Prostate cancer is an extremely high incidence disease and is the second highest mortality among cancers in US men. There's a clear clinical need for better risk stratification at diagnosis, and up to date, there's been a lack of prognostic molecular biomarkers. Secondly, so why should we study germline structural variants? These are defined as variants that affect at least 50 base pairs, and they can occur as like insertions, deletions, duplications. They can happen on the same chromosome or between chromosomes. And so big picture wise, they are vastly understudied relative to SNPs, mostly due to the technical limitations of their detection. Sequencing methods and computational algorithms for their detection have improved a lot in the recent years, enabling these large cohort studies. And while germline SNPs have been known to influence cancer risk and outcome, the impact of structural variants is still largely unknown. And finally, germline variants serve as great biomarkers due to the fact that they're inherited and fixed over their lifetime. They can be sequenced through a minimally invasive blood draw, and this only needs to be done once in the patient's life, and so this can be done even before diagnosis. So the research question I'm trying to address is, can we use germline structural variants to predict tumor molecular features or clinical outcome? This serves to address the key clinical dilemma in the treatment of localized intermediate risk prostate cancer. So for prostate cancer treatment, cases with low risk and high risk have relatively clear-cut treatment options, with low risk patients undergoing active surveillance and high risk or metastatic patients undergoing localized and or systemic treatment. And it's this middle group of intermediate risk patients that there is some difficulty in risk stratification. So there are many cases where patients undergo localized treatment where their tumors were actually indolent and slow growing, and we could have saved the cost and the impact of comorbidities due to treatment. And on the other hand, there are many of these intermediate risk patients who only undergo localized treatment and later experience relapse. So our approach is a structural variant GWAS with various tumor features and survival outcomes. We're using a discovery cohort of 300 intermediate risk prostate cancer patients, of which we have extensive clinical annotation, as well as paired tumor normal whole genome sequencing. And so with this information, we perform some univariate association analyses, and then go on to validate them in external cohorts. Yeah, so our discovery cohort is 300 localized intermediate risk prostate tumors. These patients have undergone definitive local therapy and are hormone naive, so they have not undergone any systemic therapy. Again, we have whole genome sequencing of the tumor normal pairs, as well as clinical data. So structural identification or structural variant identification is done through the DELI algorithm, which uses paired end and split read support to detect these variants. Previous methods for structural variant identification relied largely on these array based methods where we compare the relative abundance of probes placed throughout the genome So these are good at picking up large scale deletions and duplications. However, they do not have very high resolution and also cannot detect balanced rearrangements where the copy number does not change. So with newer technologies, we have short read and long read sequencing. And so for short read sequencing, which is what we are using and what the Delhi algorithm is based off of, it uses the fact that in Illumina short read sequencing, we have these paired reads where we know how far apart these reads should be. And so when we detect a pair of reads where the distance between them is far greater than expected, as in this figure, that's how we can call a deletion. And similarly, a single read can also be split across this deletion. There's also long read sequencing, which is probably the best method for structural variant identification, but currently methods are either too expensive or have high error rates. So yeah, the structure of the study is a genome-wide association study done between structural variants and recurrent somatic alterations, clinical features, or survival outcomes. We're performing Bonferroni multiple testing correction, as well as the following tests for the various feature types. So for dichotomized or categorical data, we're using Fisher's exact tests, Spearman correlation for continuous variables, and log rank tests for time to event survival data. So moving on to the results. Within our discovery cohort, we found an average of 2,700 structural variants per patient, with around 17,000 total across the entire cohort. 6,000 of these were detected in greater than 10 patients, and these are the ones that we proceeded with the analysis for. And here's just a scatter plot showing the concordance between the minor allele frequency of the structural variants that we detected compared to the NOMAD. It's a publicly available database of germline structural variants in a cohort of, I think, like 10,000 genomes. And here we see a fairly good concordance, so this gives us some confidence in our calls. And so moving on to the results of the various association analyses. So we identified, so of the 6,000 common germline structural variants, we identified 48 candidate SVs that were associated with at least one clinical or molecular feature. These are represented by the columns in this multi-panel plot with their covariates on the top and the bottom. So we have the minor allele frequencies as well as information about their location and variant types. So on the left, we have the various features that we tested against. So there are survival features as well as clinical features and recurrent somatic alterations, as well as some global mutation density counts. So in the middle will be a dot plot where the size and color of the dot will represent the effect size, and the background shading will indicate significance, with the black being statistically significant and the gray being 1 order of magnitude relaxed to account for unresolved linkage disequilibrium. So here's the various results, and what we're most interested in is mostly these survival characteristics, which serve as direct measure of their prognostic value. It's a biomarker. And giving an example just to visualize what what some of these can look like. This is a Kaplan-Meier plot, which shows the estimated proportion of patients with each genotype of this specific deletion. And this represents the proportion of patients in that group that have not experienced the event of interest over time. And here, the event of interest being biochemical relapse over 36 months. Here you can see for this particular variant, the survival outcomes are well stratified across the genotypes. So moving on to the validation, the main publicly available data set for cancer comes from TCGA, which is the Cancer Genome Atlas. And here there's 494 prostate tumor normal pairs. They also have, or they have the clinical data that we need to perform the similar associations. But unfortunately, the data is only whole exome sequencing, as well as a SNP array. And as I mentioned earlier, structural variants are difficult to detect without whole genome sequencing, as many of the breakpoints lie outside of coding regions. So you really need coverage of entire genome to identify all of these breakpoints. So to address this issue, we're using similar methods to other GWAS methods where they leverage reference panels to impute missing data. So this is similar to when we use SNP arrays to impute genome-wide SNPs, as well as when we use SNP data to impute gene expression, such as in transcriptome-wide association studies. So here, the model of choice that we are using, we want to use a model to enable structural variant GWAS in cohorts that don't have whole genome sequencing. So there are many cohorts, with whole exome or SNP array data, which will provide much larger sample size and power for either discovery for other methods or for validation. And the method of choice you're using is a random forest model to predict structural variant genotypes from SNP data. So here we use our discovery cohort of 300 patients as our training set. And we validate these models in external cohorts where we have whole genome sequencing data with both SNP and SV calls. So these external cohorts can be used for this validation, but not for the primary validation, as we don't have somatic or clinical data for these patients. So if it's just germline whole genome sequencing, but we can use that to validate the random forest predictions. And so to give a bit of background on what is random forest. So this is an ensemble machine learning algorithm that aggregates many decision trees to perform classification. Random forest can also be used for regression, but I'll be focusing on its use in classification. And as an ensemble of decision trees, it's also important to understand what are decision trees. These are binary trees that repeatedly choose splits among the training data features that best separate the class labels. So here, the class labels being the structural variant genotypes, and the features being the various SNPs. Some pros and cons of decision tree-based methods. They're highly interpretable, as the logic in them, it's like similar to human decision making. They can also capture combinatorial Boolean relationships, such as XOR relationships, that regression-based models cannot. But the major con of decision trees by themselves is that they heavily overfit to the training data. When we grow deep trees, we can achieve extremely high accuracy, or in other words, low bias, but this comes at the cost of high variance, where the models do not generalize well to data outside of the training set. So that con is the primary motivation behind the random forest method. So in a random forest, it uses bootstrapping to generate many independent decision trees, and then aggregates the results to achieve better model generalizability. So this reduces the variance of the model with a slight cost to bias. Another advantage of random forest is that it comes with implicit cross-validation due to each decision tree is trained on a subset of the samples that are sampled with replacement. So not every sample in the training set is used to train each individual tree. So for each sample, we can use all of the trees in which it wasn't used to train that tree, and then we can test the accuracy there. So this is similar to cross-validation, and this concept is called out-of-bag error rates. So we trained random forest models in the 6,000 common SVs in our discovery cohort, using all the SNPs within 1 megabase pair of each breakpoint, as well as having at least an R-squared correlation with the structure variant of at least 0.2. We remove SNPs with low diversity or ones not found in TCGA due to the fact that our goal is to apply these models to TCGA, so we need the features to be present there. And finally, the implementation uses the random forest R package with 1,000 trees, and the number of features subsampled for each decision tree being the square root of the total number of features. So next, to discuss how we are validating these models. So as I mentioned before, we have out-of-bag accuracy metrics. This provides internal validity metrics, but does not give you information on how the model generalizes outside of the training data. So here we are using external cohorts. We have 2 external cohorts, which we have whole genome sequencing data, as well as the corresponding SNP and structural variant calls. This is just a quick overview of our data sets. So here you can see discovery and validation has whole genome sequencing, but TCGA does not. And a big issue that we ran into when trying to do this validation is the fact that not all of our data sets were run on the same reference genome build. And I'll highlight some of the consequences of this in the next few slides. So here on the left is a density plot of the minor allele frequencies for the structural variants in the discovery cohort. And then in order to do the validation in the other cohorts, we need to take the SV calls that were done in GRCH37 and map them to GRCH38, and then perform the regenotyping. And here you can immediately see that the distributions are not similar. And in these scatter plots, this issue is much better highlighted. So here, when we do the mapping, we see 2 distinct subsets where one set has strong concordance across data sets, but there's this second subset of variants where the frequency that they're detected in the GRCH38 cohorts is much lower although it's not 0. So there's something happening here with the mapping, which we are investigating. And so just like from before, when we do the GRCH37 comparison between our discovery and the publicly available Nomad, we had strong concordance there. And as well as if we compare between our two GRCH38 cohorts, there's also strong concordance there. So this is highlighting some potentially interesting artifact in trying to map structural variants across different genome builds. And so going into some of the performance of these models, I'm also going to take this as an opportunity to highlight the importance of choosing performance metrics when your data set classes aren't balanced, which is frequently seen in genomic data. For example, with these structural variants, most of them are going to be homozygous reference, with less being heterozygous, and then very few usually being homozygous alternate. So here, this is a confusion matrix where you have the actual condition and the predicted condition. On the diagonal are your true positives and negatives, and the off diagonals are your false positives and false negatives. And so this figure is from Wikipedia, and it's a great resource for comparing the various performance metrics that are used. So all of these are basically different combinations of these 4 values. I'm not going to go over all of them. And the ones that I'm going to focus on are some of the commonly used metrics, such as accuracy and the F1 score, as well as the Matthews correlation coefficient. So here, accuracy is probably the simplest metric, very easy to calculate, and just uses the number of correct predictions divided by the total predictions. And then F1 score tries to account for the performance in the various classes by using harmonic mean of precision recall. And then the MCC has that complicated formula from before, but it's essentially the correlation coefficient between the observed and predicted classifications. And so the implications of these will be more apparent in the next few slides. So this is the out-of-bag performance of our training cohort. And you can see by all 3 of these metrics, does very well. The random forest models do very well in predicting within the training set. However, when we come to so this is the data for the first validation cohort. So here we expect poor performance in at least a subset of these due to the issue with the mapping. So even though this is our test set, currently a large proportion of our test set data is not accurate. But despite that, if you only used accuracy or even the F1 score, you don't really see that problem, and performance seems decent. But if you use the MCC metric, which is generally recommended for heavily imbalanced classes, this issue becomes really apparent. So we have good performance in the well-mapped variants and poor performance in the poorly mapped variants. And we see similar results in our second validation cohort. So the next steps, or what I'm doing currently, is unfortunately due to this issue and to try to get to the bottom of what is really happening here, I have to redo the discovery in GRCH38. These samples, whole genome sequencing data is extremely large, with around 500 gigabytes per sample. So redoing the processing of these is really expensive, time consuming. This is probably like the dry lab version of waiting for your mouse models to mature or something. yeah, so once I have that data, then I'll be able to better characterizing this SV mapping issue across genome builds, since we'll have calls from the same data set done in both genome builds. And then finally, once we can validate the random forest models, then we can apply these to TCGA whole exome data to get the SV calls and then validate the original associations. So to summarize, the question that I'm trying to answer is whether we can use germline SVs as a molecular biomarker to predict tumor features and patient outcome. And this addresses the key dilemma in the treatment of intermediate risk prostate cancer. So far we have identified 48 germline SVs that are associated with some of these features. But we're still working on a more robust validation. And finally, a byproduct of this whole process is that we'll have implemented and demonstrated efficacy of a random forest model that can enable structural variant genome-wide association studies in cohorts without whole genome sequencing. And with that, I would like to acknowledge the numerous members of the Boutros Lab, as well as my funding sources, and happy to take any questions now.